Hi, this is Brian Johnson, and thank you for downloading another episode of Device Talks. It's the diagnosis we all live in fear of. A cloudy spot on the MRI or the CAT scan leads to a diagnosis of brain cancer, or glioblastoma melanoma, stage 4 brain cancer. It's a death sentence. The 5-year survival rate is minuscule. The amount of medical care is tremendous. Surgery to remove the tumor, chemotherapy, and then the tumor grows back, and you have to do it all over again. It strikes the young, primarily, and up until now, there's really been very little hope for people with this diagnosis. Today, we're lucky to talk with Bill Doyle. He's the chairman of NovoCure. NovoCure actually has a treatment through the FDA for patients suffering from this condition. It's providing some new hope for these folks. NovoCure has developed a treatment called tumor treating fields, or TT fields. They're low-intensity alternating electric fields that disrupt cell division through physical interactions with key molecules during mitosis. Essentially, it takes the cancer cell and kills the cancer cell using electrical fields. Currently, it's being used in conjunction with chemotherapy, standard chemotherapy, and showing some very good results in terms of improving quality of life and even longevity of life. As some of you who may have had a history with loved ones with this disease, you'll know that any extra time you can get is precious and incredibly valuable. And that's why we were interested in talking to Bill about NovoCure and about what they're working on, because there's some very exciting developments that they have in the pipeline. Brain cancer is just the first solid tumors that they can have an impact on, and we wanted to hear all about it. So we sat down with Bill last week and had a terrific conversation, and I hope you enjoy it. And we're recording this intro just 24 hours after learning that my favorite musical artist, Prince, passed away at his home near Minneapolis. So rest in peace, Prince, and your music will live on forever. Thank you for downloading this episode, and I hope you enjoy. So the treatment that NovoCure has developed, this is a treatment that's for what most people would consider the sort of worst news you can ever get, right? You have stage 4 glioblastoma brain cancer. I'm going to take this in sort of two parts. Now, we'll go to the tumor treating fields and how Dr. Palti came up with that, but I'd like to find out how you first were drawn into this. This is not your background, correct? The oncology space?
Well, I was trained as an engineer, and when I first met Professor Palti, I was working for Johnson & Johnson, and I was in the business development function, and I was traveling around the world looking for interesting potential technologies to bring into to J&J. I actually spent quite a bit of time working on a project that was created by one of Professor Palti's students. It was in the cardiology area, and met Professor Palti during that time. So I had an expertise in the use of medical technologies to apply life-saving therapies. But you're right, oncology was not my specific expertise. And about the time I left J&J around 2000, Professor Palti had begun to work on this project, and I'm sure he called a lot of people, and I was one of the people that he called. And I think as a result of my engineering training, I understood the concept. I knew, at sort of the basic level, what an electric field was. I understood the theory at the time of how electric fields could be tuned to enter cells and interrupt cellular division. I was very impressed by the initial both in vitro and animal data that Professor Palti had generated, and determined that by no means it was a sure thing that it would work, but that there was enough evidence at the time that it made sense to take the next step.
So what kind of person is Dr. Palti? Is he the kind of person that comes in and says, I have this unbelievable breakthrough here that you need to see? Or is it more of a soft pedal thing? I mean, how excited is he about the breakthroughs that he has when he comes to you, and how far along was he?
So, I think I can describe Yoram Palti as an unqualified genius. He is a true expert, as I said, in the effects, in many things, but certainly in the effects of electrical phenomenon on the body, electrophysiology, not just in cancer, but as I mentioned, the first project that I became familiar with was in cardiology. But I would describe him as one of these guys who is incredibly inventive. If you look at almost anything, and this can be, the pen you're writing with, or the recorder that you're recording this conversation, and if Professor Palti thinks about it for 15 minutes, he's probably coming up with a better way to do it than we're doing it today. Now, not every one of those ideas is commercially viable. Some of them will take 25 years and 25 billion to develop. But he's one of these guys who is always thinking based on first principles. How can we do something different and better than we're doing today?
Right. Do you recall how he ended up focusing on cancer cells in the brain and brain tumors specifically? Well,
so he didn't start out with brain cancer specifically. So one of the interesting things about tumor treating field therapy, that's what we call this therapy, and we haven't talked about it yet, but we're using low intensity alternating electric fields. And for many people that's not intuitive, but magnetism is intuitive. So often I use that as an analogy. If I have a magnet and a piece of iron, something called a magnetic field, it's invisible, we can't see it, but that magnetic field will pull the iron to the magnet. An electric field is not the same. Some people confuse them. It's not the same, but it's analogous. If I have a plate with a positive charge and say I have a particle with a negative charge, an invisible force field that we call an electric field will pull that negative charge to the positive plate. They actually exert a force on that negative charge particle. And that force is what we are creating inside cells with our equipment, and we're using that to push and pull charged proteins that are necessary for cell division, and by pushing and pulling them out of place, rather than the cell dividing nicely, one cell becoming two becoming four, which is, the problem with cancer, uncontrolled cell growth, that one cell now dies during the division process, and one becomes zero through the application of the electric field. So it's a very general mechanism, and in Palti's initial work, he showed that it worked on all the cell lines. He actually had a laboratory that he set up in the basement of his home, and it worked on all the cell lines that he's tried it on, and it's worked on every cell line that we've tried it on subsequently. We wound up in the brain really through our interaction with the FDA. When you bring something completely new and completely unexpected, there's always questions, right? And there's always proof that is required. And some of the questions here were, will this affect the heart? Will this affect the GI tract? Right. Will it have effect on other other tissues. And so it was really through the influence of the FDA that they said, okay, we want you to start up in the brain, and we want you to start with the very sickest patients. Again, this is not unusual for something new,
right.
But that's really how we wound up with GBM, which was as a way to avoid the possibility of other side effects. Now, I will say right now, all of those concerns have since been determined to be not issues. We treat lung cancer. We have open trials in ovarian cancer, pancreatic cancer, mesothelioma. So we are treating tumors of the trunk and abdomen with no systemic toxicities. But in the very beginning, that was a question.
Yeah, absolutely. And does he bring a prototype to you, or is it still a hypothesis? I mean, you're the first. I think I read you were the first professional investor in the company, so I assume he had to have been a little farther along than.
Yeah, so I was not the first investor. There was a very knowledgeable angel investor named Leonard Perlhagen, who was a former pharmaceutical executive who made the initial investments. But yes, at the time that I invested, as I said before, there was extensive in vitro data, so petri dish data, and he'd also done the very first experiments in animal models. And what I really, my investments led to the first human clinical trials. So that was the gap that I helped to bridge.
And then you ended up as chairman of the company. Was that?
Yeah, it turns out that this is one of the most, if not the most interesting project that I've been exposed to in my career. Here's an opportunity to create a new class of therapy. So when we think of cancer therapies, we obviously think of surgery, we think of radiation, and we think of pharmacology. Clearly, lots of advances being made, particularly in the pharmacology area. But this is truly a fourth and compatible modality. It's a physical modality. It has not been anticipated before Palti's work, and it has the potential to be used broadly in some of the most deadly cancers. And the team that Palti had recruited was an outstanding team. They had an outstanding CEO, Asaf Danziger, an outstanding chief science officer, a gentleman named Dr. Alon Kirzon, an outstanding chief operating officer who made sure all the equipment works and is delivered, a gentleman named Mike Ambrogi, and many others. But one of the challenges always at the leadership level for these companies is that there's a conflict or a division between the time that's required to actually accomplish what needs to be accomplished to move the project forward, and the time that's required in order to finance the project, represent it publicly, set the overall strategy. And Asaf Danziger and I developed a wonderful working relationship, 2003, 2004, it's now over a decade, where we decided to divide and conquer that responsibility. And it's worked out marvelously for the company. So that's how I became the chairman, and how we split the leadership role at NovoCure.
Right. So let's step back for a moment here, because I realize we haven't actually sort of described what the device, the Optune device that you have currently. So, I mean, if I look at it, I think we initially years ago kind of said it was analogous to a shower curtain, a shower cap that had electrodes in it. But that's very rudimentary. But why don't you describe it in terms of how you describe it to people when they ask you what you do?
Sure. So let's go back to when I was describing the basic therapy. So the idea is to apply these electric fields to the region of solid tumors, so the tumor itself, and also the region surrounding the tumor, so that to the extent the tumor has spread within the region, we can fight the tumor there too. And in order to create those fields, we need to. I'll go back to the notion of electric field between two plates. So we're obviously not using two metal plates, but we use what, for all intents and purposes, look like bandages. So in the case of brain cancer, there's a bandage that goes on the left side and the right side of the head, and that creates one set of opposite transducers, and then there's another set of bandages that go front to back that create the second set of transducers. And then those bandages are connected by a wire to a field generator. The current field generator, our Generation 1 field generator, is carried in a backpack by the patients. It weighs 6 pounds, and the patients use this. It's a wearable device, and they use it all the time. Essentially, when it's on, it's killing cancer cells, and when it's not on, it's not killing cancer cells. It seems to be a magic number around 18 hours a day, that the patients who use it 18 hours a day or more have a much better survival benefit than the patients who use it less than 18, although 17 is better than 16 and 16 is better than 15.
Sure.
But 18 is what we really shoot for. We have a second generation device because one of our goals, of course, is to continue to make this easier and more manageable for patients and their caregivers. Our second generation device reduces the weight by more than half, so we go from 6 pounds to 2.6. We no longer need the backpack. It now goes into a purse or in an over-the-shoulder bag. And that is now approved and rolled out to our European patients, and we've filed with the FDA for approval in the U.S. And our hope is by the second half of this year we'll be rolling out the nice small lighter device to the U.S. patients as well.
Right.
And I would expect over time, batteries keep getting better, that we'll continue to make improvements in convenience as we apply our engineering to the problem.
Right, and I saw on your website, most of the patients have to have a shaved head, correct? And the bandages are very, very thin. And I mean, I saw one young man who was wearing a baseball cap, and you could barely tell. So the bandages are like bandages, and the patients will typically replace them 2 to 3 times a week. They do have to make intimate contact with the skin in order to transmit the electric fields to the tumor region. And so as the hair grows during the week, or as the patients exercise, they will change the transducers. This is all done at home. They don't need to come to the hospital or back to a clinic. Either a loved one or many of the patients are fully capable of doing it themselves. And of course, if it's pancreatic, which is not approved, that's a trial, or ovarian, which is also in trial. It's the same thing. The bandages, the transducer arrays are bigger because we're covering a bigger area, but it's the same thing. You stick one on your back and one on your abdomen, and then one on each side. Mm-hmm. So TTF, you're calling it essentially the fourth field in fighting cancer. The first three are, as you said, I think you said, pharmaceuticals, surgery, and chemotherapy.
Well, I try not to get too pedantic about that because right. I have had people, say, "Oh, but what about vaccines? Isn't that the fourth?" So it's clearly a new, novel therapeutic modality. Whether it's the fourth or the fifth, we don't need to argue about that.
Sure. I mean, I guess my question is, it could be an incredibly dumb question. What is it about the current standard of care that has left this opening, you think?
Well,
besides
clearly,
that, ultimately, these are a lot of cancers people are dying from. But I mean,
yeah, I think it's as simple as that. I think that we have learned, particularly over the last 20 years, a lot about cancer. We continue to learn a lot about cancer. But therapeutically, there are very few cancers still, particularly the solid tumors that tend to be discovered late, where surgery is not curative, where we can offer patients, an extended long-term survival. That's what the whole body of researchers and people who are focused on this ultimately want to provide. And it appears for many of these cancers, the answer is a combination because these cells are so clever, if you will, about evading various interventions. And I may not have emphasized this early on, but we're not focused on eliminating surgery, radiation, and chemotherapy. In fact, we've shown that Tumor Treating Fields works particularly well with chemotherapy. Depending on the type of chemo, we always see at least additivity, meaning 1 + 1 = 2, so you're always better adding. And with some chemos, and most notably so far the taxanes, which also attack the same cellular mechanism, we see profound synergy, which means, 1 + 1 = much more than 2, and in the case of our laboratory experiments, we see an increase in sensitivity to things like taxane by 1,200 times. So there's a real opportunity to, we believe, improve the care of patients with with solid tumors, and we can talk about our data in glioblastoma in a minute. Importantly, too, we do not increase the systemic toxicity with this therapy. So often when you add two cancer therapies together, there may be an improved survival benefit, but many times there's also, if you will, an enhanced toxicity. We don't see the enhanced tox. So you get the therapeutic benefit without, as far as we've been able to discover in our work thus far, any systemic toxicity penalty.
Right. So, while we're here, why don't we just touch on some of your data, because I think it does kind of lead into other discussions. So you just study the results of one of your studies. The EF14 trial was released in December, published in JAMA. Congratulations, that's very good right there. But
and that is our most important trial to date. Yeah. So, as I said, when in our initial interactions with the FDA, we determined that for all kinds of reasons that we were going to focus on glioblastoma. This is the most common primary brain cancer. It has a terrible prognosis. There's at best a sort of single-digit 5-year survival using standard therapy, and the mean overall survival is somewhere in the 15-month kind of range. So
we'll go there.
It's among the most difficult cancers.
Absolutely. And the study that you released, it was 700 patients, correct? And it was basically it said that it measured, I'm not going to be able to pronounce the oral chemotherapy, but it was the oral chemotherapy in combination with the Optune versus just the oral chemotherapy alone. And you see it showed significant increase in both progression-free survival and overall survival compared to just using the oral chemotherapy alone. I just want to kind of unpack the statistical differences in terms of this trial. And I guess we should note that this trial is measuring people who are initially diagnosed with glioblastoma versus recurrent, correct?
Correct. So, if I, maybe chime in. So our belief because this therapy, compliance is important, as I mentioned. We want these patients to use it at least 18 hours a day for as long as possible. There's no systemic toxicity, and it works with the chemo. We believe that the best therapeutic outcomes would occur if they use it at the time of diagnosis. So that was the design of this trial, was to prove or disprove whether that is correct. These patients typically present, or we're all not getting MRIs every year to check for brain tumors. So these patients will typically present with a seizure or some massive headache or some neurological deficit. You know you have a problem. The patients get an MRI, and if the MRI shows what looks like a tumor, a mass, these patients will typically get surgery almost immediately.
Yes. Within weeks of the diagnosis, right? I mean,
exactly. So these are the patients. You get an MRI on Friday and you're in surgery on Monday. If it's confirmed GBM, then the standard of care is to receive radiation and low-dose temozolomide. I'll pronounce the drug for you.
Thank you.
I've learned to pronounce it. And that they'll receive that radiation. It's typically the measurement they use is gray, 60 gray, and they'll receive that over a period of 4, 5, 6 weeks. At that point, they then, under standard therapy, will begin to receive higher doses of temozolomide. And that is the point where we started treating. So the control group received the standard therapy, which were the normal course of temozolomide, and the treatment group received exactly the same thing, but in addition, they received the Optune therapy. It's important to note that they continued to receive Optune therapy through progression, and that the treatment group, the control group, also received second-line chemo after they progressed. So it's a very simple trial. In concept, the control group received standard therapy, and the treatment group received standard therapy plus Optune. You mentioned that 700 patients. We actually recruited 695 patients from 80 centers globally. And like most of these big oncology trials, there's something called an interim analysis, and this is a statistical look in the middle, if you will. And the reason for these interim analyses generally are for futility, so that if it's clear in the middle that the therapy is not working, the idea is not to put another cohort of patients through another several years of recruitment and therapy. Our interim analysis was performed 18 months following the 315th patient. And a couple of important, maybe historical notes. The last successful phase 3 trial in newly diagnosed glioblastoma was the temozolomide trial that's now over a decade old. And there has never been a trial that has been successful at the interim. That changed. So and the data at these interim analyses, the data don't come to the company. They go to what's called an independent data monitoring board. Our data went to the independent data monitoring board, and we received the call from this group that consists of a statistician, an oncologist, and I believe a neurosurgeon, that in fact, surprisingly to everyone, we were dramatically successful after the first 315 patients. So we had demonstrated statistically significant superior progression-free survival.
So that means that the tumor doesn't grow back, right?
Yeah. There's actually a very specific criteria that's used. This is evaluated by independent radiologists, so they receive a batch of MRIs, and I believe the criterion is that the tumor has to grow about 25% to be considered recurrent. And as I said, that's evaluated independently. And that is the definition of progression-free survival. It's a very interesting statistical measure because, again, it's completely independent of the investigators. But if you're a patient, what you care about is the second endpoint, which of course is the overall survival, which is the time and the improvement in survival of the patients. And that too was statistically significant and superior.
I think what I'm looking at here is overall survival was 19.6 months versus 16.6 months. Does that sound about right?
Well, the actual FDA endpoint, and because this has been presented in different fora along the way, but the FDA endpoint, the control was 15.6, and the treatment was 20.5. Okay. So it was about a 5-month increase in the median. Numbers that are also very important to the statisticians are the hazard ratio, which was very low at 0.66, and we all know about p-values. The p was 0.004. But I think in terms of patient numbers, the number that I find the most encouraging are things like the 2-year survival. So the 2-year survival in the control group was 32%, and in the treatment group it was 48%. So you go from about a 30% 2-year survival to about a 50% 2-year survival, and we continue to follow these patients, and over time, we will publish the 4-year survival and the 5-year survival, and of course the hope here is that we will show significant improvements in those longer term measures as well. But those are to be reported.
Well, that's excellent. I'm wondering, in terms of historically, how does that compare to early studies on like the oral chemotherapy or initial treatments in terms of what they showed in terms of overall survival rate? Is there any way to compare how statistically significant that roughly 5 months more on a median basis is?
Well, again, that is the control group. So the control group is the standard of care. So that's the nice thing about a phase 3 trial, that you really are comparing apples and apples here. So again. We don't have to look back historically because we have an actual control group in the trial. So I think the 15.6 to 20.5 is the number, and 32% 2-year to 48% 2-year survival is those are the real numbers.
Yeah. So I guess I'm just trying to gauge in terms of how big of a leap this is forward for the treatment. I mean, maybe it's impossible to sort of get there, but I'm just sort of wondering how to put. Well,
I guess there's some things we can say, which is again, in over 10 years, there's been no improvement. Yeah. At all, and there've been many different attempts. I think too, our hope is that as we introduce the new device that I talked about, the smaller, lighter device, that will allow more patients to have more, achieve the 18+ hour a day therapy, and continue to do better. So we're really happy with these data. These are the best data. Best phase 3 data that have been reported in this incredibly difficult disease. But our ambition is to continue to improve our therapy and continue to offer even more hope to these patients.
Yeah, and I certainly didn't want to make it sound glib, I mean, the people who get glioblastoma, I mean, they're statistically rather young. Aren't they not? I mean, they're fighting for any amount of time they can get usually to be with family and loved ones and live as much of their life as possible.
Yeah, so GBM is not a geriatric disease generally. Yeah. It hits the median age is in the 50s, sort of the mid-50s. So this is a disease that typically hits in the prime of life. And remember that the data that we just talked about are the medians, right? So that means half the patients have done worse, and half the patients are doing better. And we do see this long tail. And we're studying that tail to, for all the clues that we can figure out with respect to, and again, the one that we see is compliance. The patients who use it the most do the best. But these are the things that offer the opportunity for further study and to allow oncologists to really coach their patients to get the maximum benefit.
And when you received your FDA approval for TTF for recurrent GBM, they included a quality of life claim, which is unusual, correct? I mean, is there? Can you unpack that a little bit?
Sure. So let me take one step back, which is again, I think the most important data that we've published and the data that were published in JAMA in December we just talked about. But when we got started, as I said, there were lots of different concerns with the new therapy. So the FDA required us to start with sicker patients, so not the newly diagnosed patients, but the patients who had already failed radiation and chemotherapy and whose tumors were progressing. So the first trial that we did was a very simple trial. Half the patients were receiving salvage chemotherapy, and half the patients were receiving Optune monotherapy. So without any additive chemotherapy. These patients were very sick. 10% approximately were first recurrence. 90% were second, third, fourth. I think we even had sixth recurrence patients. So again, these are patients who, were really along the disease path. And in this trial, what we showed was that again, going back to the medians, the median survival was the same between salvage chemo and Optune monotherapy. It was about 6.6 months for the Optune and 6 months for the chemotherapy. That's statistically the same, even though the Optune's a little longer. But we showed some other very interesting things too. The first thing and by the way, that was the basis of the claim, which was equivalence to chemotherapy with superior quality of life. Because again, we didn't show any of those systemic toxicities that you typically see with chemo. Okay. I should mention there is one side effect for Optune. I don't want to say that there are none. The side effect that we do observe is in a fraction of the patients, around 40%, we see a contact dermatitis under those bandages. So if I put a bandage on your head for 2 years, there's some chance that you're going to get, the redness. And that's typically treated with, over-the-counter creams and things like that. It almost never interrupts the therapy, but that is the side effect that we see. But going back to this trial, the couple of other interesting observations was, number one, we saw a dose response. So this is where we really observed that the patients who used it 18 hours a day or more actually did better than the chemotherapy patients at all points on the curve. And if you used it less than 18 hours, it was better than nothing, but you did worse than chemo. So, again, this points to the fact that what sounds obvious, but it has to be on to work. And then the other thing that we saw, even in these very sick patients, in the control group, there were essentially no survivors at 3 years. Whereas in the group that received Optune, there was a percentage, about 8% of the patients, of all the patients who were alive at 4 years. And again, these are patients who were expected to have, very few months when they started. So there was a cohort of very long-term survivors. So that provided the basis of this initial indication, but really the clues or the real indication of promise that gave us the motivation to do the EF-14 trial that has been so successful.
And so, the difference. What's the hypothesis in terms of the difference of starting the treatment as soon as you are diagnosed versus having to wait until it's recurrent? Do you think there's going to be a significant increase in overall survival, or are you not really allowed to speculate like that?
Well, we're seeing it, right?
Okay.
Because the overall survival in the recurrent group was 6 months median, and the overall survival in the newly diagnosed group was over 20 months. Wow. And again, we were measuring that from the time we started therapy. So it's from the time of diagnosis, it's actually a bit longer than that. Yeah. And the hypothesis, at least my hypothesis, is that the patients can use it longer. They're better able to keep up with the therapy. This is a therapy that you have to change your batteries, you have to plug in, and healthier patients are much more able to derive the benefit from the therapy because they can keep it on.
Yeah.
I think it's almost, there's no doubt there's more complexity than that, but, I think that's a big part of it.
Sure.
And also, by treating it at the beginning, we can treat it with the chemotherapy, where we've shown a benefit of using the two therapies together. There's no question that the two together are better than either one alone.
Yeah, it's incredibly exciting breakthrough, and from what you're telling me, I wonder if there's ever the challenge, not to try and boil the ocean when you have this technology that's going to work across all these spectrums. And I wonder just, how do you guys manage your own expectations? How do you stay one step at a time, or is it easy to do that? Well,
I'd say that we are, as a company, have two tracks. Track one right now is to provide Optune therapy for all the GBM patients who, with on-label can benefit from the therapy. And that means training oncologists, it means training centers, it means manufacturing equipment, it means training patients, and we're really focused on that every single day. We believe that this is the best therapy available for GBM patients. We believe that based on our data. It's not based on a gut feel, and we really want to make sure that the patients who can benefit have it available. And so that's sort of track one. Track two, which is where I think you're going, is that as I mentioned, we've seen this work on every proliferating cell line that we've tried it on in the lab, and we have a long list of various cancers. Yeah. Our limitation is not a mutation or a histology. It really goes back to physics. We need to be able to surround the region with our transducers. So we're not doing blood cancers. We're not doing cancers of the bone marrow, but basically the cancers of the head, the chest, and the abdomen, which include many of the of the worst solid tumor cancers, are our targets. So here we are. We're still an early stage company, but we're trying to be as ambitious as we can. So one area that makes, we believe, a lot of sense are brain metastases. So GBM is primary brain cancer. It's actually cancer of the brain cells. But there are many more brain cancers, that actually travel to the brain from other places. Lung cancer, breast cancer, melanoma, are among the big ones. Mm-hmm. And we're in the process of initiating a clinical trial to treat brain mets, which are very difficult to treat from other cancers. Mm-hmm. We have very good phase 2 data in non-small cell lung cancer, and we're working to determine what the best phase 3 trial, where we can contribute the most in non-small cell lung cancer. And I think one of the most exciting things that we've described recently in January is initial phase 2 data in pancreatic cancer. At a meeting called ASCO GI in January, we presented data where we added tumor treating fields to the standard of care chemotherapy, a drug called gemcitabine, and it's phase 2 data. We don't want to. And with phase 2 data, you look for promising signals that will lead you to a phase 3. But we saw very promising increases in survival, and very promising increases in the response rate. So the number of tumors that actually shrank while they were under therapy. So, and again, we know pancreatic cancer is another one of these most difficult cancers. So we're trying to do. We can't do everything. But we're trying to focus on these very difficult cancers where we think we can make a real contribution.
Well, that's terrific, and it's exciting to think of all those cancer cells in those petri dishes, so to speak. The treatment working on all those. So I can't wait to keep following the story and see where you guys are going next. That's all the time we actually have. So I wanted to ask one final question. You're working with these people with glioblastoma. What have you learned from dealing with these patients? Have you learned anything about character, about life, about survival, and anything you can share with us?
Well, this is one of the great privileges of this project, and at the end of the day, we are absolutely focused on the patients. We didn't talk about our business model, but we send technicians to every one of our patients to deliver the equipment, explain how to use the equipment, make sure that they're compliant. So we have a very direct contact with our patients, and this is, on the one hand, a devastating disease, but on the other hand, the character, the determination, the tenacity of these patients is inspiring, and it is really the reason that, not just me, but I think every employee of NovoCure is so dedicated to making progress as quickly as possible, because these patients are such tenacious people, and we really want to provide them with the best therapy that we possibly can. And we want to provide it quickly, and we want to provide it in a way that allows them to derive the maximum benefit from the new technology.
Well said, and I wish you the best of luck in that. Thank you so much for joining us.
Okay, my pleasure.